Good day, this is the Professopher, CBMD PhD. And today's Friday, December 16, 2022. It is 3:13 and 39 seconds, AM Eastern Standard Time. And today's research, skin cancer. So let's do it here. Activated vitamin D3. Treatment may reduce the risk of arsenic-mediated skin cancer. Health trial for activated vitamin D3 may protect us from the harmful effects of arsenic in drinking water. Date December 15, 2022. Source, Shabora Institute of Technology. Millions of people across the globe regularly consume arsenic-contaminated water. Exposure to arsenic has previously been associated with the development of various cancers, including skin cancer. Research on the underlying molecular mechanisms regulating arsenic-mediated carcinogenesis remains sparse. Using in vitro studies, researchers demonstrate how carcinotriol for activated vitamin D3 inhibits arsenic-mediated carcinogenesis in certain types of skin cells known as keratinocytes. According to recent estimates, over 140 million people from 50 countries regularly get exposed to arsenic through drinking water. The exposure level significantly exceeds the guideline value, 10 microgram liter, stipulated by the World Health Organization. It is an established fact that chronic arsenic exposure from drinking water causes a variety of cancers, including skin cancer. Unfortunately, there is a general paucity of data on the underlying biological mechanisms that regulate arsenic-mediated carcinogenesis. Moreover, methods for the prevention and treatment of arsenic-mediated carcinogenesis have remained elusive thus far. And these cells are primarily found in the epidermis, the outermost layer of the skin. It is a scientifically established fact that certain signaling molecules, kinase proteins, e.g., MEK or AKT, that control the fate of various biological processes, are strongly associated with tumor development. The in vitro study in human non-tumorigenic HAC-AT skin keratinocytes showed that calcitriol, which is also known as activated vitamin D3, 125 dihydroxyl vitamin D3, inhibited arsenic-mediated anchorage-independent growth with downregulations of cancer-related activation of several signaling pathways, including MEK, ERK1/2, and AKT, as well as activity of cell cycle. And to elucidate the relationship between arsenic uptake and carcinotriol treatment, the researchers measured arsenic levels in HAC-AT cells, long-lived, spontaneously immortalized human epidermal keratinocytes treated with carcinotriol using an inductively coupled plasma mass spectrophotometer. Quite interestingly, arsenic levels in HAC-AT cells cultured with arsenic significantly decreased when these cells were treated with increasing doses of carcinotriol. The carcinotriol significantly repressed arsenic uptake in HAC-AT cells with the regulation of expressions aquaporin genes AQP7, 9, and 10, which were modified by arsenic exposure. The vitamin D receptor expression was significantly increased by arsenic exposure, whereas carcinotriol had no effect on the expression of the receptor. The researchers then sought to understand whether calcinotriol had an inhibitory effect on arsenic-induced tumorigenesis in cells other than skin keratinocytes. And to this end, they performed anchorage-independent growth assays using a human normal lung epithelial cell line called BIOS-2B. The results of these assays were equally astonishing. Arsenic-mediated anchorage-independent growth of BIOS-2B cells treated with carcinotriol was suppressed by 21.4-70%, suggesting that carcinotriol's potential to suppress arsenic-induced tumorigenesis is not restricted to keratinocytes. And these results suggest that carcinotriol suppresses arsenic-induced tumorigenesis not only in keratinocytes, but also in other target cells, including lung epithelial cells. And furthermore, the expression pattern of aquaporin genes evolved arsenic uptake, a critical step in arsenic-induced carcinogenesis, is significantly altered by carcinotrial treatment. Researchers therefore believe that activated vitamin D3 or calcinotriol may contribute to the prevention and therapy for arsenic-mediated diseases, including cancer. Environmental toxins, such as arsenic, contribute significantly to the development of life-threatening diseases, such as cancer. However, it may take years, even decades, for cancer to develop from drinking arsenic-contaminated water. The current research clearly indicates that calcitriol could be used as a test compound for validating the safety and efficacy of activated vitamin D3 and/or its analogs in preventing or treating arsenic-triggered cancer. And by taking vitamin D3 beforehand in arsenic-contaminated areas may reduce the risk of cancer development 5 or 10 years later and help people maintain good health for a long time. This is certainly welcome news for millions of people forced to survive on polluted water worldwide. And this is the Professopher, Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.